I want to present you some of the applications and the developments that we're doing at the Laboratory for Optics and Biosciences, and I will mostly focus on FLIM and metabolic imaging, although we have many different types of projects with harmonics, but I don't have time today. So with second harmonic generation and third harmonic generation. So for metabolic imaging, I think you had also an introduction about phasor analysis by Enrico and by Suman. So I'm going to go very briefly with this introduction. I just want to remind you that metabolic imaging and with microscopy endogenous biomarkers has a subcellular resolution, which is very has many advantage for measuring subcellular components in cells. And also, of course, is label-free because we don't need any external label, and we are using endogenous biomarkers. As you probably already know, we have a lot of endogenous biomarkers that are very important for physiological processes. And among them, we have NADH, FAD, retinol, retinoic acid, lipofuscin, keratin, melanin, and porphyrins as examples. This list is not exhaustive, of course. And as you see that, as you know, they have very broad absorption spectra and emission spectra. And that's why one of the reasons we want to perform fluorescence lifetime microscopy, FLIM, because not only we can identify the type of molecules we have through the radiative decay, but FLIM is very sensitive also to the microenvironment through the non-radiative decay. So we can have information about bonding, quenching, and pH. So I'm just going very quickly here because I know you had very extensive lectures. I just want to say that most of the data that we're presenting today are acquired with time-correlated single photon counting, so in the time domain. And then we measure the fluorescence decay in every pixel of the image. This is the pictures of one of the custom-made microscopes that we have for FLIM. And we are performing analysis with phasor analysis, which is fit-free and assumption-free. I'm sure Enrico and other people gave you a lot of details about that. I just want to remind that one of the power of phasor analysis, beside being fit-free, is also that it is linear in terms of lifetime components and molecular species, so we can quantify the ratio of molecular species in a graphical way. And we have like a references that we started doing when I was back at LFT with Enrico, in Enrico's lab, like a mapping of endogenous, the fingerprint of endogenous biomarkers. So we can use this as a reference. So we are mainly focusing for the metabolic imaging on NADH and FAD, because these are related, they are involved in the energy production and ATP production in the redox states of the cell. And as you know, NADH and FAD are the primary electron acceptor and donor in oxidative phosphorylation. So in this in the mitochondria, and NADH is also involved in glycolysis in the cytoplasm. So since the pioneering work of Britt and Chance, the optical redox ratio was initially evaluated by intensity ratio by FAD or over FAD plus NADH, which is, of course, can work. But as you know, like intensity measurement can have a lot of artifacts depending on the intensity, excitation, variation of absorption, especially in tissues. So FLIM is lifetime, measuring the lifetime is much more precise because it's independent on the concentration and variation of the excitation. And as you know, NADH lifetime has a short 0.5 nanosecond lifetime when it's free and long lifetime when it's protein bound. And especially the lifetime depends on the type of enzymes that is bound through the cycling in the TCA cycle. So NADH has been used a lot, especially for quantifying glycolysis and oxidative phosphorylation. FAD is much less used but holds a lot of potential, and this actually holds or is like an active research field. So just very briefly, FED has a short lifetime when it's protein bound and long lifetime when it's in the free form. So if we go back to the NADH, we can make two schemes about, one cell that has more oxidative phosphorylation, that has a high bound to free NADH ratio, but a cell that undergoes mostly to glycolysis has a low bound to free NADH. So what in practice has to do, as you already saw, we can measure the lifetime of NADH in every pixel and quantify with a Phasor analysis the fraction of bound NADH, so the ratio between free and bound NADH, with this graphical analysis, and quantify the ratio between glycolysis and oxidative phosphorylation. And we actually started doing this while I was a postdoc with Enrico at the LFT. And then here I can show you some examples of the paper that we together. We can map metabolic gradients inside tissues. And I just want to point out that, as people already did, that it's very important to do metabolic calibration inside the cells, for example, with metabolic effector, by blocking the respiratory chain. For example, we see like this movement in this metabolic trajectory toward the free NADH, or inducing oxidative stress, we increase the bound NADH concentration, or we can change glucose concentration or inhibit glycolysis to see where is the metabolic between glycolysis and oxidative phosphorylation. So after this brief introduction, I want to talk about what we're doing at the Laboratory for Optics and Biosciences. So we are performing here some hardware development, in particular with Xavier Solinas. We developed a custom-made, lab-made electronics based on FPGA to perform FLIM, and then we can adapt this for the sensitivity and speed and the number of channels. The current version has 2 channels and a temporal gate of 500 picoseconds, or channels, but we are working on improvement both on channels and the temporal resolution. So on the other side, we are also working a lot on the analysis, and in particular, we started during the pandemic and the lockdowns to develop software, which is called Fluorescence Lifetime Ultimate Explorer, FLUT, which is custom written and is going to be free, and is going to be open source, and everything is written in Python by Dale Gottlieb, and we have been testing and implementing this with all the FLIM team, like the postdoc Lin Ung and another PhD student, Bahara Sadipur. And this is going to be, again, as I said, published soon on GitHub. And one of the main features is that we not only can do like cluster selections of the Phasor analysis, but we can very easily map the phase contrast or the modulation lifetime, or also the fraction of bound NADH in this case, or other fluorophores, in a very interactive way, and then we can perform masking and analysis in the FLIM images. So everything will be published on GitHub. If you want to have more details, you can just come back to me to our lab. So I want to talk about 2 main projects. So we have many projects on metabolic imaging. So the projects I will talk about first is multicolor 2-photon excitation of the endogenous biomarker, and to characterize also the sensitivity of NADH and FED in long UVA-induced metabolic stress. And another project is in collaboration with Lorena Aguilar Arnal, the UNAM, National University of Mexico, to study metabolism and lipid synthesis during the stem cell differentiation. So starting with the first project, what is the motivation? The motivation is that many papers in the recent years show that the 3 different biomarkers, like the optical redox ratio, the NADH lifetime, and FED lifetime, can give very different information that are kind of complementary. still is not fully understood what are the metabolic pathway that are reporting on, but you can see like some examples from Melissa Scala's lab and Ari Georgioukoudis' lab, in which they use different types of metabolic biomarkers, and they can distinguish different types of metabolic fingerprints and phenotypes of cancer cells and stem cells. So this is actually very important and very interesting, and we want to develop more this multiparametric imaging. But one of the limitations is that if you want to perform simultaneously NADH and FAD imaging with one wavelength is kind of complicated. So although they have overlapping spectra, the concentration of the two biomarkers is different. So it's very hard to get like a good balance of the two intensity in the two biomarkers simultaneously. So what people do is usually excite at around 750 and excite in the blue for NADH, and then they move to 900 and excite in the green for NADH. This is of course possible, but for very dynamic system, you have moving artifacts. It's very hard to get the same image in the same region of interest. So so we started to develop multicolor, multiphoton microscopy with wavelength mixing. So the wavelength mixing has been first developed at the LOB in like 10 years ago, basically, for rainbow applications. The principle is that we have two laser beams, so lambda 1 and lambda 2. Lambda 1 is exciting a blue fluorophore and lambda 2 a red fluorophore. And if we overlap them in time and space, we create a virtual wavelength that is exciting the green fluorophore in multiphoton, with one photon from one laser beam and one photon from the other laser beam. And so this technique is intrinsically co-aligned because we have the two foci that has to be overlapped. And we adapted this technique, wavelength mixing, to the image NADH and FAD simultaneously in tissues. So we use the commercial laser with one fixed output at 1040 and one tunable set at 760. And when we overlap and synchronize temporally the two beams, we create this virtual wavelength around 900 that allows to excite NADH independently on FAD. And as you can see here, when we synchronize the two beams, we have a good FAD contrast and intensity simultaneously on with the NADH intensity, something that we don't have if we use only one wavelength. So this is very useful for very dynamic system. This is just a proof of principle of multicolor with wavelength mixing. And I just want to point out that this is, of course, compatible with second harmonic generation or third harmonic generation. So here we can, in second harmonic generation, we can see the microtubules during cell division. But we apply this technique mainly for imaging reconstructed skin. And then here we have the first demonstration of one-shot NADH and FAD, so optical redox ratio and lifetime. So we can explore the heterogeneity in these three different parameters. And then we can also map metabolic gradients in redox ratio, NADH lifetime, and FAD lifetime that are associated to differentiation. But the main application I'm going to talk about today is a collaboration with L'Oréal and in particular with the team of Ana Maria Pena. And the goal is to study the sensitivity of NADH and FAD to metabolic stress induced from a long UVA. So long UVA is longer than short UVA and of course UVB. So is, let's say, has less visible damage, but is still inducing oxidative stress. This is much less studied. So what we have is we induced UVA treatment with a solar simulator, which is a UVA lamp. And then we image our reconstructed skin before the treatment, 30 minutes after the treatment, and 2 hours after the treatment. And we use physiological doses of 25 joules and 40 joules. And what is very important is that we want to look at changes in metabolism at early stages, so 2 hours. So at this stage, 2 hours after exposure, we don't have any morphological changes, but we still have oxygen, reactive oxygen species. While after 2 days, we have apoptosis and cell dying, both in the epidermis and in the dermis. So what do? We image our skin sample in physiological conditions, both at the basal layer, where we have the stem cells of the epidermis, so keratinocytes, and then we go also in the dermis, where we have fibroblasts embedded in collagen. So what we observed after UVA exposure, we observe an increase of NADH lifetime that corresponds to an increase of bound NADH, or NADPH in this case, and an increase of FAD lifetime that corresponds to a decrease of bound FAD. And then we believe that this is really associated to the oxidative stress and the response to oxidative stress. So here you have a gallery of examples, both in the epidermis, in the basal layer, and in the epidermis, in the fibroblast. What is happening in control and different types of treatment with different doses. We see that we have an increase of fraction of bound NADH, a decrease of fraction of bound FAD. And then if we pull together all the data we have over hundreds and hundreds, almost 1,000 of cells, we can observe that we have very interesting, the metabolic response is biomarker dependent, but also cell type dependent and dose dependent. So in fact, we observe that the NADH is showing an early modulation in fibroblast, but not in keratinocyte. FAD biomarkers showed an early modulation in both cell types. And the FAD is very interestingly, is also sensitive to the UV dose in the epidermis layer. So this is also probably due to the different antioxidant response of different types of cells. And then I want to briefly talk about another application that we've been working on. And so this is a collaboration with Lorena Aguilar-Arnal at the UNAM. And this has been funded by the Human Frontier Science Program, and the goal was to study the interplay between epigenetic and metabolism during stem cell differentiation, with a particular focus on SIRT1, which is redox-sensitive transcription factor. And for the FLIM development, we actually developed metabolic imaging of NADH and lipid-associated fluorophores with the subcellular resolution during the maturation of mesenchymal stem cells into adipocytes. Then we combined this FLIM measurements with other measurements like seahorse analysis, PCR, RNA sequencing, and also pharmacological treatments to inhibit or enhance the SIRT1 activity. So coming back to the microscopy, so here we performed all the microscopy images with custom-made, is a semi-commercial and semi-custom-made microscope from La Vision Biotech. And here we can do FLIM and also THG. So to excite NADH and lipid-associated fluorophores, we excite at 740. But the lipid-associated fluorophores have a very large spectrum and is actually not really known. So we use third harmonic generation microscopy as a ground truth to visualize the lipids and the, for people who don't know what is THG, is a harmonic contrast, is a third order process that is actually sensitive to the variation of Chi3. And in practice, in biology, you can see the interface between water and lipids. So if you look at the THG intensity in the adipocytes, you can clearly see the lipid droplets and the border between the lipid droplets and the water. And so where the lipid droplets are located, we see a long lifetime of the autofluorescence. And this was also previously observed by Rupsa Datta, the LFD, and also another lab, George Coudy's lab. So we use the ground truth of CHG to set the thresholds for the lifetime and to automatically select the pixels and attribute them to the lipid mask and perform automatic segmentation at the pixel level. So we can assign pixels to lipid mask or to the NADH mask automatically in every cell. And then we can apply this to our measurements. So here we have stem cell during differentiation into adipocyte, day 4 until day 15. So this is the fully developed adipocyte. And with different treatments, this is the control, this is the activation of SIRT1, and these are inhibition of SIRT1. So this is the intensity, this is the tau modulation. And then if we perform our lipid automatic segmentation, we can quantify this with the lipid ratio, so the pixel of the lipid mask over the total pixel. And then we can see that over time, we have an increase of the lipid ratio. And if we look at NAD plus treatment, we have smaller lipid ratio. So we show that NAD plus is actually hindering the differentiation process and the lipid accumulation. So we tested this technique over also more traditional oil red staining. And then finally, so the last part, so if we look at the NADH mask, we can quantify the fraction of bound NADH over the differentiation, then we can see that we have an increase of fraction of bound NADH that is actually showing a metabolic shift from glycolysis to oxidative phosphorylation. This is kind of expected during differentiation. And then NAD plus treatment is actually decreasing the fraction of bound NADH, so is decreasing the oxidative phosphorylation. Moreover, we did subcellular segmentation of NADH based on intensity. We know that NADH intensity is much higher in mitochondria, so we use a threshold on the intensity to separate the NADH in the mitochondria and assign them to mitochondria mask. And below these thresholds, we assign the pixels to the cytoplasm and nuclear mask. So by using this subcellular NADH segmentation, we can show that the subcellular compartmentalization of NADH actually depends on the cell state. And then here, the day 8 of development, we see that if we compare the controls to the inhibition, we don't see any difference. But in mitochondria or nuclears and cytoplasm, we see like a difference. So we have a compartmentalization and different things that are happening at the metabolic level in different compartments. So now I want to conclude. So in this talk, we show that we can perform label-free, non-invasive imaging, multiphoton imaging and FLIM combined with also harmonic generation. That NADH can report on OXPHOS, oxidative phosphorylation, glycolysis, and oxidative stress. And then we can perform multiparametric imaging of NADH and FAD simultaneously by multicolor two-photon microscopy by wavelength mixing. And with this, we can do one-shot NADH, FAD, FLIM simultaneously. And this, with this technique, we demonstrated the metabolic responses to UVA. We demonstrated that this is biomarker, cell type, and dose dependent. And then we can establish like a new methods to quantify lipid biosynthesis and subcellular NADH metabolism inside the cells.